Thank you, and thank you for the invitation. I think my main goal today is to get everybody excited about the technologies and biology. Biology is always at the basis of what I'm talking about. So I'm a molecular biologist by training, but 14 years already I went over to the dark side and I started to dabble in a little bit of things related to policy and regulation, and then also helping bioinnovation in South Africa. So the background there is, what is Biosafety South Africa trying to do? We're trying to support bioinnovation for the building out the bioeconomy in South Africa. As you know, anybody that's been involved and heard anything about biotechnology, it's not it's not a cheap technology to commercialize. So there's cost, expertise, compliance issues, and acceptance issues. And we are involved in all of those different issues. So the role of a platform, we're funded by the Department of Science and Innovation. The role of platforms is to reduce those challenges for innovators. And then another thing that I will talk a lot about today is about sustainability. And what do I mean with sustainability of bioinnovation? It's basically about these four different components. If you look at the biosafety aspects, and when we started this organization, we only focused on the biosafety, and that's why it's called Biosafety South Africa. The biosafety is the health and food safety issues, and then the environmental issues, but we very quickly realized, and it's actually formally regulated in South Africa as well, as the so-called socioeconomic issues, or I like to divide it into social political things and the techno-economic ones, because techno-economics are generally things that are fairly, I wouldn't say it's simple, but it's at least easy to identify and handle these things. Techno is what I'm going to spend a lot of my discussion on today, because I want to let people at least appreciate a little bit of what this technology is, the basis of how it works, and then what the potential applications is. And that's the economic side. Biotechnology is about developing products. And products, you only have products if you have a gap in the market for those products. And then the socio-political things is the things that I referred to earlier as the dark side a little bit. We need a policy framework in which to do these things, and it's regulated technologies, so we also help people with this. And then all four those components, if a product complies to that, we refer to it as a sustainable biotech product or sustainable bioinnovation. All of this is fundamentally based in responsible governance. And for that, you need the policy and the regulation, as I already mentioned. It's pretty straightforward, but also technical capacity. And I think this is one of the challenges that we have, and it's one of the roles that we have, is that we take the hands of policy developers and regulators and bring in some of the technical expertise that they require. So the technology and the product. So this section is quickly going to try and give you a few examples of how the technology works and what it's based on. And I want to start off with this slide just to say that it's a complex environment. And there's a really important reason. It's not to say, this guy works in a complex environment. Who does not? But the point is, every term that you use has a potential impact on the implications or the meaning. So when I start talking to a technology developer and I use a certain term, that person may understand something completely different than a policymaker. And things, in Afrikaans we say, the pop falls on the ground between the hand and your mouth, basically. But that's what happens. Things get lost in translation, often. So we need to pay attention to these things. And this is why I bring this up. Today, the two that I underlined for you there, new breeding technologies, and in genome editing, you will hear, in the end, I'll probably only refer to genome editing because that's what I'm used to. New breeding technologies is a term that we developed in a very early stage, and we spoke about these things because they were new. But you can understand that you can never keep on calling a group of technologies that, because obviously new things become old in the first place. And later I will explain to you how badly defined it is. But unfortunately, this is still lingering in the policy environment, so we still talk about new breeding technologies. And I will focus on this complicated slide a little bit later, but I just quickly want to highlight, I just told you that we talk about these technologies. And if you just have a look at right at the top left-hand side, well, let me go one stage back. Right at the top, you will see I talk about techniques. So if we talk about regulating this technology, what I realize is we have to define three areas. The first one is the techniques that we use that I try to do, and then you will see there are blocks to try and group these things. Then if you look right in the middle, the blue box, under the arrow there says genetic impact. So you use some tools, techniques, you have a particular genetic impact, because we're going to talk about why obviously we want to change genetics. Everybody that works with seed know this already. And then right at the bottom you have application. So again, this is one of the complicated issues. I can use sometimes the same technique to have completely different genetic impacts. So when I start talking then about safety issues, it may have the same technique may have completely different impacts or risks associated with them. And then the same with application. So when you do risk assessments of these technologies or of the products of the technologies, then you have to actually look at all of these different things. So that is important. And then what you have to do, and this is what all the debate is about, somewhere that big gray arrow that points from the left to the right, that's a very vague indication of change in the genetic or the genetic impact. So if I go back to right at the top left, you will see there I actually included something like breeding. I tried to put in these older technologies as well to give some context when we start talking about genetic engineering and all the things. And like I said, I will talk about it later. But coming back to the red line, then we regulate these things in addition to all the other regulations. Remember when you bred a plant and you do the cultivar registration or whatever, you have to look at things like genetic stability. You have to look at obviously things like allergens and the food safety issues in any case. So these technologies or the products are in any case regulated. But if you use genetic modification, for example, it's in addition regulated by other stuff. And this is the line that I'm trying to draw there. This is a very important part of the discussion today because one of the international discussions that's going on is, should all the products that we develop, so first we just did breeding, then we started to do in vitro breeding. I actually should explain it in my next slide here, and start at the beginning. Natural, we see variation naturally. Everybody knows about this, depending on which mutations give that organism a particular competitive advantage, those will be retained in the population. The whole population will actually drift into that direction. Then we started to understand breeding. So what the main thing there is, you select the two parents that have the traits that you look at. Then we started to look at things like in vitro breeding, where we introduce things like polyploidy, triploids, hexaploids that you work with. We could actually use high energy radiation to induce mutations, and then we select the particular traits that we have. So it's all about changing the genetics to get phenotypes in the crops that you work with that you want. Then we started to talk about genetic modification. So what is the big thing about genetic modification? In a simple way is, for the first time, we could actually isolate genes from sexually incompatible species and transfer them. Bt maize is a good example. We isolate the gene from a bacterium and you put it into maize to give you insect resistance. And today we're going to focus predominantly on what I call just genome editing or gene editing. So I will try and explain in a little bit later how it's different, but already some of those products are on the market as well. So first thing, let's talk about the technology a little bit. And what I'm trying to do here is just to show you in a very simplistic way on a genetic level. There you see the two chromosomes, a blue one for the male and a pink one for the egg cell, for the female part. And then you have an embryo. Usually you have a little bit of segregation of traits that come out, but you basically limited to the genetic variability and the mutations that happen during this whole process when you do breeding. When you do in vitro breeding, you actually induce, and what I try to show here is now we, for the first time, actually started to specifically try to change the genetic material. We didn't really know what we did. They literally put seeds into a container and they expose it to radiation or to chemicals. And then you plant out the seeds, the embryos were then mutated, or some of them, and then you select from that big mixture of phenotypes that you get from that, you will select if there's anything that you want. I explained this already. There you can see I put in a purple bacterial or a bacterium there, and from that we isolated a very specific sequence, a gene, and we incorporate it into a cell. And now for the first time, look at now we have suddenly a purple flower that started off as white. So it's a completely unique. So it's still a very powerful technology that we shouldn't discard of completely. And what I try to show here now is genome editing focuses much more on the genetics of the plant. So you do not necessarily bring in from something externally, but you change the genetics that you induce the phenotypes that you want. So the last one I then want to show you then is this, again, just summarize. So the important part of this slide is look at the 4 little cells that I have at the bottom there. Is what are these genetic changes that eventually translate into a phenotypic change? So they basically, you can categorize them in mutations that I indicate there with a star. You can have rearrangements, and you can have inserts, and then you can have deletions or insertions. So there's a reason why I show you this. If we induce that, it's exactly the same as what we did. Previously, we were just dependent on what happened naturally. Those were the mutations, the type of mutations and changes that occurred naturally, and we kind of just waited for them and then we selected. You can understand that it's a slow process. So inducing the mutations at least gave us more variability, but obviously you realize that that variability could be something, even in some cases, which we didn't want. Then we get to genetic modification. Again, there's my insert. You can just see it a little bit clearer on this slide that's bigger. And now we come with genome editing, where we have tools that you can actually now identify a very specific area in the genome and make a change there that you want. And now it starts to become a little bit complicated because you can see there, genome editing, I refer there in the first one I just had there, the acronym SDN. It stands for site-directed nuclease, and I will explain it in the next slide. But you have different types. The 1 and 2 inserts only a small change, if we call it, or we can insert actually a whole gene. And this is why I put it in the middle of. So you can actually use genome editing to insert a new gene. So we cannot, if we go to regulators, or regulators should not tell us, let's regulate genome editing in a particular way. Because genome editing products can be highly variable. This is what I'm trying to show you here. And there's my little tick marks. If I look at what happened naturally, what happened with my induced mutations, all of which these technologies and the products are not regulated in addition under the GMO Act, for example, in South Africa. And I can do now exactly the same thing, but because of the mechanisms that I use now on the right-hand side, that mutation I can actually target to a very specific sequence or a gene in this. So it's a much more accurate way of doing it. So I want to emphasize the point that there's theoretically no difference between those products. But still we're discussing about should we regulate these. The other thing that's important for breeders, those first 2, like I already explained, you do or you wait for the mutations or you induce mutations, but then you have to plant the seeds and see what you actually got from there. So it's a selective, reactive way of selecting then what you want. Whereas when you design these things with genome editing, you can actually proactively design the mutation and insert that into. So it's a huge advantage. So coming back to this complicated thing, let's now focus into what I call then the new breeding techniques. The first thing that you will see is that some of them are listed under the techniques, some of them under genetic impact, because these are actually what you read there. The site-directed nucleases that I have there, and then in the blue at the bottom, things like cisgenesis, intragenesis, all those things are lumped together as the so-called new breeding technologies, just purely because they were new at some stage. But we talk about completely different things, and this is why the details matter. So quickly to try and explain to you one set of genetic engineering, it's the CRISPR-Cas system, which you all have heard about. And I think there's only 2 things to say here that's really important for you to understand. It's looking at that top picture, you will see in the blue, that's your target DNA. This is the organism. This is the soybean or the wheat genome that you want to edit. Then you have this CRISPR-Cas complex, which is the green in the first place. It's a target, it's an RNA strand that you actually design, and you know that DNA or nucleic acids in general, RNA, also have complements. So this is what gives you the specificity. So you can design a target RNA that will go to a very specific, and something like any genome in the human genome, for example, you need only 11 sequences to identify a unique sequence. So it means that if you make engineer a CRISPR-Cas system, you can take what is in what orange color there is a protein, and that protein is a nuclease. So a nuclease cuts the DNA into 2 pieces, and that's what they show below. So first, let's just quickly again emphasize what is that CRISPR complex, it's two things. It's an RNA that gives you the specificity, and it's a protease that gives you or induces the cut in the DNA. So what you end up then, you will see there on the left-hand side, you have cut DNA, and then they refer that to NHEJ. It's non-homologous end joining. So what it means there, you just literally cut the DNA. And remember, this happens naturally as well. So the cell mechanisms, there are enzymes that will actually fix the DNA again. So it's part of the mechanisms, the natural mechanisms in the nucleus of all living cells. But when they rejoin the DNA, it makes mistakes every so often. This is one of the reasons, for example, we develop things like cancer. If that mistake induces proliferation and cell growth, we see it in the end of the day as something like, like cancer. So by introducing that mutation, you can change the DNA. So you can, what they refer to as an indel, it's a small change which can completely take out or knock out the gene, for example. On the right-hand side, it's exactly the same thing, except there's now a break, but what you put as part of your CRISPR-Cas complex, you also put in a new sequence with border sequences on the edges that are homologous to your target. So they will be able to hybridize on the left-hand side and the right-hand side of your break, and the piece in the middle will be now your new insert. So it means now also I can do, this is why SDN3 that we spoke about earlier is about inserting new genes, but I can now put that gene in a very specific way, or I can duplicate genes next to each other and all those type of things that I want to do. So there's SDN1 on the left, SDN2 and 3 on the right, as I indicated. And then just quickly again, this is a very simple overview. In the meantime, also research and development does not stand still. They have now changed because we start to understand how these things work. It's like any other technology in the world. We can now start tinkering around with those things. So we can make it more specific. We can do things like they have now base editing and prime editing and all those type of things that you can just change. You don't even induce the double straight breaks. You only put in a single strand, lots of different things. So the technology changes. So here's then the exciting part, the opportunity, and I always like to just throw in this thing there, is to say the examples that I'm going to show you is only what other people have done before. Don't let your own creativity be limited by what other people did. Theoretically, biodiversity is based on genetics. If we can change the genetics, there's huge potential for us out there. So non-browning mushroom, 2016, the first product in the US that was commercialized, very simple, polyphenol oxidase, the thing that causes the mushroom to go brown, apples to go brown. It's a reaction by the plant cell to protect it against insects, for example. We don't like it, especially if we're a little bit lazy and we quickly want to make a risotto with mushrooms in the evening. We don't want to first chop the mushrooms. We want to buy white ones off the shelf. There's the high oleic acid soybean, 2019. It's probably the second product that went commercial. Gluten, low gluten wheat that everybody that suddenly has, what do you call it? I forgot the protein complex now. Sensitivity, that it's away. And then biofortified tomato. You see, lots of examples. Those were the plants. They were really think about, have you ever thought why some cattle have horns and some not? It's a genetic mutation. So if we understand the mutation, we can induce it. If we already have some meat or whatever breed of cattle, we can induce that same, still have exactly the same breed, but without horns, because we do only that small genetic change. And then, of course, as always, the biggest investment and the biggest research and development goes into the medicine development. There's huge things. This was several years ago already. It was twins that had leukemia when they were born. And long story, but they did some genetic engineering on some of their stem cells, replaced it, and they were cured. So then a deep dive on one particular crop. I have maize here. It's just to show you quickly. At the top, I have a lot of traits that are changed. So this, just quickly, one step back again. Why is this important to me? If you think about genetic engineering, GMOs, we only have 3 crops in South Africa, 3 traits. Only insect resistance, herbicide tolerance, and drought, which is a latecomer in any case. So why is it? I will show you a little bit later. It's hugely expensive. It's complicated, all those type of things. Now we have a technology. I'm actually giving you my conclusion now. But we have a technology that can really make these changes that we want to. So look at the top there of maize. These things has already been published. So we can change the characteristics of the corn itself. We can then look at things like the more traditional things that we did in GM, the agronomic traits to make life easier for the farmers. And then right at the bottom, you can introduce actually traits that can help you with your breeding program. So the world is your oyster, as they always say. This is another example where they call it a de novo domestication of wild tomato. So one of the big issues that people talk a lot about, especially if they talk about these heritage things that you can buy off some shops' shelves, is that the loss of biodiversity. Ooh, we have all of these commercial things, which coincidentally the consumer wanted, but now there's other values in the system, so we have to adapt to that. But now you can actually change, and you can quickly go from a wild type, much more quick, and the genetics behind that, I am honest, I do not understand completely, but at least you can come from a wild type and go to a commercial viable cultivar much quicker. This is a company that we worked with a little bit, that were in South Africa a while back. So canola and the alfalfa, are examples of just a single gene, that DHS, some dehydrogenase gene, which I can't remember exactly, but it's a single gene that they downregulated. And look at the impact that it has on architecture, for example, and the growth, which then has phenotypes that we obviously would like. The alfalfa on the right, lucerne is obvious. And then just quickly, a quick look at what have people done already. Remember, again, this is in the context of GM, where we have worked now for 30 years with only a handful of products on the market. This is looking at research publications that have a direct application topic. And there you can see 59 different crops have already been edited. That's the relative numbers. Rice is never a surprise if rice is number one, because it's such a huge staple, and the research infrastructure and stuff in the countries where it's a staple. And then all the others. There's soybean number 3, canolas 4, wheat is actually 4, and then canola. So a lot of work. That's the point of this slide. And a variety of crops as well. Again, coming back to the commodities that we have as GM. There's the traits. Again, I'm very excited about this. I already gave you some examples, but just look at the variability of traits here. I will come back right at the end of my conclusion slide. I will come back to why it's so important that we look at different traits, for example, I think. The technology that's used, predominantly the CRISPR-Cas. I just showed you CRISPR-Cas. Remember, there's a lot of other technologies. The first ones that were commercialized were actually not CRISPR-Cas. It was only discovered subsequently, but they used things called talents, which are huge proteins, or those are the transcription factor type things, which can also bind specifically to DNA. This is why it falls in the same category. And then on the right, it is you remember our old friend SDN1, 2, and 3. The point I want to make here is I showed you, hopefully, early on, that SDN1 is like things that happen naturally. So we should not regulate it as GMOs, in my opinion. And it means that 99% of the innovation that's going on at the moment should not be regulated as GMO. So you should get excited about that as well, if we can just get the policymakers and the regulators to see it the same way. Countries, again, China, USA, it's just interesting stuff. And the big question, this is one of the reasons why we also exist, is where's South Africa? It's completely absent at the moment. So we need to do, and again, remember, this is the first thing that I want to do, is I want to support local innovation, particularly small companies, but obviously we help, and we've helped everybody with the regulatory and the compliance issues. So, quick comparison then, just the large circles tell you something about the top right is innovation diversity. So it's who's actually doing the innovation, crop diversity, and then trade diversity. And then I just compare this, so it's not apples with apples, I do agree, but genome editing with genetic engineering in South Africa. So in the first place, diversity of innovators, all the commercial stuff is multinational big companies. We want to support local innovation. Crop diversity, only the 3 that I referred to. Look at the diversity, and I already showed you in a different way. And the same for trade diversity. So I'm just repeating what I said earlier in a little bit, but I always also contrast it to the genome editing. And this is then just to, again, remind you quickly, this is only about 2 months old, this publication, but GM development, 16 years, 1.7 billion rands. I know that we can do it at 10% of that cost in South Africa, but still, a big company, that is what it costs. So the point I want to make is genome editing, much higher productivity because of the simplicity of the technology, the targeted focus. There's lots of reasons why it's quicker through the development pipeline. 90% small, medium enterprises friendly, again, which may be relevant to some, and then the diversity. So regulation, just quickly, we've debated until last October what we should do. By that time, all those countries that I've highlighted it has made those many decisions. Unfortunately, we can't look at this anymore because a country like US and Australia, for example, already said they're not going to regulate SDN1. So they will never, again, we can't go to the regulators and say how many SDN1 products you have because they will not have any documentation on it. Our government decided based on the definition, so it's not a technical, it's not a risk-based assessment that they made. They made internally, without the input of a single attorney, they interpreted the definition in the Act. So, again, just a different way of showing variability induced from left to right as natural breeding, etc. The point I'm trying to make is, again, that SDN1 is even better than natural selection. But we have decided to regulate it. It's not fixed in stone, I believe. We're still fighting that fight, so we'll get there. This is the point I made earlier. This is on top of the other regulation that we have in any case. People often think when you say it shouldn't be regulated as a GMO, they say it's a free-for-all. It's not what we mean. We just say it should not be regulated. So at the moment, if you look globally, there's green and red places. It's only South Africa and New Zealand at the moment that decided to regulate SDN1 as GMOs. European Union is doing it currently, but it's based on a court ruling, and they're busy revetting. I'm feeling positive that they may be a little bit more enlightened after that. I've spoken to some of my colleagues in regulation in Europe, and they are not as optimistic as I am. One of the issues that you will appreciate more than I do is how do we regulate this in the end of the day if we go this way? Because in the first place, a single nucleotide change, you will not be able to develop a detection system like we have done previously with GMOs. And if we sit on an island, it's just not going to work. So my conclusion is that genome editing is really, truly, I believe this, it's an opportunity for the rest of Africa. I go into other African countries about biotech regulation and frameworks and things a lot, and they want this even more than we do. And why do I say it's an opportunity? Is because technically it's much quicker, better. The time, it's reduced, the cost. I mean, who can afford 1.7 billion rands? And then risk barrier is reduced for the reasons, hopefully, that I showed to you. So we need to complement it with sensible regulation. So this is the slide that I said. My concluding slide is to say, how do we realize this? The policymakers should come to the party. Our GMO Act, the regulators should actually change. Why do I give this to you? Most of you people will probably say that's the most boring stuff in the world. But if you have influence, you must help us change people's minds. The research development system, what I refer to as the more, the people in the universities and stuff, they must come to the party. Bio businesses must invest in it. And then in the end of the day, the market is the point that I made earlier, is that we must make products that the end consumer can see the benefits for. And then we will not have these discussions about GMOs or whatever. So we must be confident about bio-innovation. It's not, like we almost make excuses why we support it. That is just to say there's some information. Our website is on there. If you have any questions, you're more than welcome to discuss it with me. Thank you.